Hello to anyone who is watching this video. This is the first in a series of videos I'll be making about interesting topics in both genetics, evolutionary biology, and oncology that intrigue me. This is mostly for my benefit, as I tend to learn very well from teaching subjects to others or presenting it in a PowerPoint format. But if anyone has any questions, I'll link the paper in the description below, and feel free to comment if you have any specific questions or other further concerns. And I think this is mostly just for my benefit in terms of my future career and going back and seeing what I've been interested in the past. But without further ado, I'll go ahead and start this video. So this video is about a potential solution to Peto's paradox, which is laid out by a paper in 2018. And as I said, I'll add this in the description to the video. But essentially, the byline of this video is, why do elephants get less cancer than us? And that really gets to the heart of Peto's paradox. So what is Peto's paradox, first of all? So Peto's paradox is named after the eponymous Sir Richard Peto. He was a famous British statistician and epidemiologist. He discovered first, he's most well known for actually discovering the relationship between cancer incidence and tobacco smoking exposure. In fact, he actually calculated along with a fellow colleague that there would be upwards of 1 billion new cancer incidences in the 21st century if tobacco smoking rates weren't declining from, I believe the 1970s or 1980s. So he was, I think, knighted for this, but he's more well known actually for his eponymous paradox. So where did this paradox come from? So there was a study that he conducted of about over 17,000 British civil servant officers that demonstrated that there was a positive, statistically significant correlation between human height and cancer incidence. And that's within, of course, the same human species, or, his cohort obviously was of the same species. But then when he extended this relationship analysis to 36 mammal species, ranging from really small, field mice, all the way up to African elephants, which are the largest living land mammal, he found that there was no such relationship between body size and cancer incidence across these species, which is interesting because as I discussed earlier, in humans and in dogs, bigger body mass means more cancer incidence. And that goes along with everything we know about cancer biology and the fundamentals of cancer development. Because we know that the more cells you have, the more chance you have of acquiring a mutation in one of those cells' genomes that causes it to turn cancerous. And this mutation risk also increases with time as well, with age. But there was no such relationship identified across species, which is interesting. So another way to think about this kind of relationship within humans, or within a single species, and thinking about this relationship between body mass index, or body mass size, and cancer incidence, we can think about two different roads. So the road on the left, we have a single lane country highway, and maybe there's one or two cars passing there in a day or even in an hour. And so there's not going to be many traffic jams along this, this lonely country road. And that equates to our, smaller animals, where they have less cells, and so they have less cancer incidence because they have less cells with which to, acquire cancerous mutations or to become oncogenic. And then going on to larger body mass individuals, we see that they have, more cells, more cars, and more incidence of traffic jams, which in this analogy is equivalent to cancer incidence. And just going back a little bit, so body mass is a function of increased cell mass because eukaryotic cells are all the same in size. So the only way to add body mass proportionally is to add more cells. Just to clarify on that point. And so in this right-sided, in this multi-lane highway with multiple intersections and bridges, multiple areas where cars can swerve and brake and cause these downstream effects where traffic slows and stops, and huge traffic jams can emerge as a kind of emergent property. We see that these more multicellular organisms, they can tend to develop more cancer incidence or traffic jams in this analogy. So that's kind of what we see within the same species, but we don't see that across the species. And here you can see a graphical illustration of Peto's paradox. We have an expected linear cancer rate, if you can see my cursor, in the orange line here, going all the way from really small animals like mice to whales. We would expect a proportional scaling or linear increase in cancer incidence with body mass because there's more cells, obviously, that can acquire mutations and become oncogenic. However, as you see the observed cancer rate, there is no statistically significant, correlation, positive correlation like that we observe, which is very interesting. So let's go on to the next slide. So just to recap everything that we talked about so far in terms of Peto's paradox, we know that mammals that live longer and or are larger tend to have lesser rates of cancer as compared to smaller species. So whales and elephants suffer from less tumor burden proportionally than humans and mice do. And again, this is contrary to the conventional wisdom that more cells and more age equals more cancer. So what are potential hypotheses to prove, to kind of fix this kind of gap in our understanding of cancer incidence across species? So potential solutions are that larger and longer living mammals may have one, enhanced tumor suppressor activity. And just to back up a little bit, there are two kind of genomic alterations that are usually responsible for cancer incidence in a given individual. Number one is the presence of an overactive oncogene. An oncogene is generally a gene that promotes acceleration through the cell cycle and promotes a proliferation and division of somatic cells. Or tumor suppressor deletion or absence or silencing. Tumor suppressors are generally genes that are the opposite of oncogenes in that they stall or inhibit cells with dysregulatory genetic architecture, and that is to prevent that dysregulatory genetic architecture from carrying forth in that cell's progeny and causing cancerous growth. So this first hypothesis has to do with enhanced tumor suppressor activity. Number two is lower metabolic rates. And this is important because lower metabolic rates generally, metabolism is generally an oxidative process. If you think about redox, your redox equations in chemistry, generally oxidation is, in oxidation you lose electrons and you produce, energy from that kind of oxidative process. And we tend to oxidate, carbohydrates to produce H2O and CO2. And that generally powers our proton gradient in the mitochondria, which is another way of producing energy through potential energy. But also oxidation produces reactive oxygen species, which are highly debilitating and damaging to double-stranded DNA and to nucleic acids in general, and cause the proliferation or acquisition of mutations. And just as an aside, larger animals do have lower metabolic rates actually. So, for example, whales, when they dive very deep into cold water, their heart rates can actually go as low as 2 beats per minute, which is very interesting to consider that their heart rate their ejection fraction is so powerful that it can sustain all that body mass and sustain all that vascularity, even at just 2 beats per minute. Whereas humans, have a basal heart rate at around 60 to 100 beats per minute. Number three is that elephants and whales have fewer total cell divisions over time. And this kind of relates back to point one, which is that because maybe they have increased tumor suppressor activity, that they proceed through less cell divisions over time, just as a more tightly controlled process. But in this paper, we're discussing the first hypothesis, which is elephants have increased enhanced tumor suppressor activity. So let's get into the paper. So this paper deals with examination of a specific gene product called leukemia inhibitory factor 6, specifically in the pay and ungulate clade of mammals, which includes things like manatees, rock hyraxes, and yes, elephants or elephantids, also called proboscideans, because they have a large proboscis or trunk. And this LIF6 protein, usually in most mammals, it is only present with one copy, but in African elephants, and also one other thing, LIF6 is classified as something called a pseudogene, which I'll get into a little bit. But essentially, African elephants have 7 to 11 copies of this pseudogene, so-called pseudogene, whereas most mammals have one copy. And just to back up and talk about what a pseudogene is, a regular gene is it has a protein coding sequence, of course, but it also has ancillary sequences called promoters or enhancers, which are usually present upstream of the gene. And these promoters or enhancers have specific receptors for other kind of targets like proteins or other kind of nucleic acids, which kind of accelerate or stimulate the protein coding function of a gene. But pseudogenes, while they do have the protein coding function, by definition actually, they lack a promoter or enhancer element, which is very interesting. How do these pseudogenes form? Well, they can form through kind of aberrant duplication during recombination, in which the protein coding sequence is the protein coding sequence, which is where mRNA is transcribed and everything, that can be duplicated without the promoter region. Or another more interesting process is where there's reverse transcription of an mRNA product of that pseudogene, the protein coding function. And remember, mRNA, it does not by definition contain any promoter or enhancer regions or introns. It is simply the exonic or the protein coding product that is transcribed from this double-stranded DNA counterpart. Sometimes these mRNA products can get reverse transcribed back into the genome and inserted back into the genome. So as you can imagine, it lacks a promoter component and it's just inserted back into the double-stranded DNA. And some viruses do express reverse transcriptase, such as HIV, classically. So that's two ways that pseudogenes can form. And actually mammalian and especially human genomes are full of pseudogenes. In fact, we may have even more pseudogenes than actually 30 to 44% of the human genome consists of repetitive retrotranscriptonic elements like this. And usually we think of them as junk DNA, but now more and more we're finding out these are actually very crucial to our evolution. Anyways, this LIF6 is very highly enhanced in elephants specifically. And actually, the authors of this article, they used something called Bayesian phylogenetics in order to reconstruct the entire kind of ungulate family tree, which obviously we know from the fossil record and other kind of auxiliary subjects, that they reconstructed the phylogenetic tree using this kind of LIF6 relationship. By seeing which, where the amplification and duplication process happened, they could reconstruct the family tree and determine how close it was to the family tree that we already know through from the fossil record and other genetic evidence. And this is very interesting. The Bayesian theory and the Bayesian phylogenetics comes from Bayes' theorem, which is where we calculate the posterior probability of an event, for example, a phylogenetic tree given LIF6 duplication data from previous data, like how well is the data actually described from what we know of the actual phylogenetic tree, and how probable is a tree, and all of these things. So it's very interesting to go back and look through. Then they looked at the actual physical architecture of the LIF6 pseudogene, and they found two things. They found that it does have amino acid sequences, specifically leucine and isoleucine residues, that are required for the pro-apoptotic function of LIF, which I'll go into later, or the LIF transcript. And that LIF6 in African elephants has a TP53 binding site. Now, TP53, if you know anything about human oncology, is a very well-characterized tumor suppressor gene that, when it's absent, causes a series of devastating cancer incidences. But it's kind of like a master switch or a master regulatory protein in that it controls many pro-apoptotic and anti-cell cycle proliferation gene products. So that was interesting. And just as a quick aside, what is apoptosis? Apoptosis is also known as programmed cell death. And this is very important because it's a way for cells that are damaged or have dysregulatory genetic architecture, or that are metabolically injured. It's a way for them to be detonated or destroyed in a controlled manner, so as to prevent necrotic cell products from leaking out into the multicellular environment, destroying other cells. Because these cells contain very highly digestive enzymes and lysozymes that can damage surrounding cells. And apoptosis is also very important in that it prevents kind of the proliferation of dysregulatory cells that have incomplete genetic architecture or insufficient for continuing through the cell cycle in a healthy manner. And so in this specific instance, there is a family of proteins called BCL2, some of which are anti-apoptotic, some of which are pro-apoptotic. In this case, we have a pro-apoptotic protein called BAX, which is targeted by LIF6, and that induces pore formation in the outer mitochondrial membrane. And that releases a specific substance called cytochrome C, which sets off the pro-apoptotic sort of caspase cascade. And so that's just a quick thing about that. So what was the meat of this kind of paper? It was a series of experiments, actually, in Asian elephant dermal fibroblasts, or skin cells. And essentially in this experiment, they were exposed to two kind of chemotherapies or DNA damage inducing substances called doxorubicin, which is an anthracycline, very commonly used in many cancers if you're familiar with that field, and an MDM2 antagonist called Nuclein 3A. MDM2 is something that promotes double-stranded DNA repair, and so antagonizing it with Nuclein 3A obviously would prevent the repair of double-stranded DNA, and that again is very damaging to cells and is a strong inducer of apoptosis. And so in this kind of experiment panel, they also use something called siRNA to selectively turn on or off LIF6, and that is to kind of delineate what does LIF6 actually do in the context of DNA damage by these two chemotherapies. So the series of experimental cohorts they had for this set were negative controls, so cells that were neither exposed to DNA damage nor had any siRNA silencing. Number 2 is exposure to doxorubicin plus control siRNA. Now this control siRNA did not selectively turn off LIF6, so this was just kind of a part of a control. Then they had doxorubicin plus an LIF6 targeting siRNA, and that is to both turn off LIF6 and to induce DNA damage, and this is kind of to see what does LIF6 depletion cause in the context of doxorubicin-induced DNA damage. Then they did the same thing with Nuclein 3A with either Nuclein 3A with a control siRNA or Nuclein 3A with LIF6 turned off with an siRNA targeting LIF6. So what were the results of these experiments? So the main thing that they were looking at was cell viability, which is another word for kind of apoptosis or cell survival, or degree of cell survival. And you can see that in this figure A here, at the very left, you can see that in the control, the cells are pretty viable at baseline, like the fold chain is 0, which means this is kind of their control setting. But then when they introduced doxorubicin here with a control siRNA, they saw a very low degree of cell viability because these cells are obviously dying from the DNA damage. But when they targeted doxorubicin damaged cells with LIF6 silencing, you can see that there's a statistically significant increase in cell viability. So even though these cells are not as viable as the negative control, turning off LIF6 seems to improve their survivability in terms of they're not undergoing apoptosis. So this suggests that LIF6 is very important for inducing apoptosis in the context of DNA damage. Same thing with, you can see with Nuclein 3A. With Nuclein 3A, obviously you have very low cell viability, but when you introduce LIF6 silencing, that viability actually increases a lot by a very statistically significant margin. And so that suggests that LIF6 is very important in actually inducing cell death, programmed cell death in the context of DNA damage. Similarly, in the apoptosis side, you can also see that apoptosis is very dependent on LIF6 in both the setting of doxorubicin and Nuclein 3A damage. So very interesting how LIF6 is actually very important for the actual process of cell death, programmed cell death, in the context of DNA damage by either doxorubicin or Nuclein 3A. So all of this is to say that LIF6 was very important in these Asian elephant skin cells in apoptosis, much more important than its kind of pseudogene status suggests. And another set of experiments, which I haven't shown here, is that they showed that this is very dependent on TP53, actually. So this is the results of that experimental course. So then this goes back to the evolutionary study. So when exactly, so we know that in humans, LIF6 is a pseudogene. It's not extremely important for us. But in Asian elephants and elephants in general, it seems to be very important for that apoptotic cascade to proceed in the context of DNA damage. So when did it actually arise? So here we have a phylogenetic tree of all kind of, proboscidean or elephantid type species, ranging from the African elephants to the Asian elephants and some extinct species like the woolly mammoth and the straight-tusked mastodon. And they essentially, what we want to do with this phylogenetic tree is look at a DN to DS ratio of LIF6. So what is a DN to DS ratio? It's also called omega. So a DN to DS ratio is very important for determining how strong selective pressure is on a gene. So DN stands for the rate of non-synonymous mutations. What does non-synonymous mean? Non-synonymous means that a mutation, this non-synonymous mutation, it encodes a mutation such that the actual amino acid or protein sequence encoded by the gene changes. And you can imagine how detrimental a non-synonymous mutation can be, because if you look at, for example, human sickle cell, you have one kind of amino acid substitution from glutamine to valine, you have a whole detrimental disease state, which I'll get into later in other videos about how that's an evolutionary kind of trade-off. But anyways, you can see how selective, how detrimental just one single non-synonymous mutation can be. And a synonymous mutation, on the other hand, is silent from the proteinaceous level, in that it doesn't encode any kind of amino acid change. And so synonymous mutations really have no kind of selective pressure against them. But if the ratio of non-synonymous to synonymous mutations is less than 1, then if you think about it, that means that this gene is very functional, because evolution is really highly selecting against any change in its protein that it encodes. And so a ratio of less than 1 signifies that this is a functional gene or a complete gene, one that has active selective pressure against it because it encodes a protein that's very important and that cannot be changed at all. But if a DN to DS ratio, or omega, is more than 1, this signifies one of two things. It could signify that because there's so much non-synonymous mutation load, that this protein this gene is undergoing strong selective pressure, because the protein product that it transcribes is very important is very kind of has a high selective pressure to it, because it is either negatively trying to be changed or positively trying to be changed. Or it could be a pseudogene, because pseudogenes, as we said, they're very kind of silent. They don't have a promoter region. Oftentimes, they don't even produce functional proteins at all. So there's no kind of, it's kind of a net neutral in terms of evolution doesn't really care if it adopts non-synonymous or synonymous mutations at all. But the authors actually found that LIF6 in the elephanted genome is under significant selective pressure. It has a very low DN to DS ratio. And so this indicates that there's very strong selective pressure against LIF6 changing its protein coding at all. And so that means it's under strong purifying selection. Evolution doesn't really want it to be changed at all in elephants. And so when did this exactly happen? When did this pseudogene, this zombie gene that was dead and is just kind of an accident of evolution or accident of transcription, when did it kind of refunctionalize in elephants to become so strongly selected for purification? And that actually happened 26 million years ago, according to the paper, right about the time when proboscideans started to adopt larger and larger body sizes. And this interestingly suggests something very important about elephant evolution and Peto's paradox. That is that there's an evolutionary cost to large body size that LIF6 seems to be solving. So essentially, random DNA sequences upstream of these inserted pseudogenes can acquire mutations to become promoter or enhancer regions. Just random mutations that kind of turn on a promoter kind of function. But there's strong selection against the de novo creation of promoters for these pro-apoptotic pseudogenes like LIF6. And that's for obvious reasons, because too much apoptosis is also detrimental and prevents body mass growth and cell proliferation to a certain extent. However, elephants tightly control LIF6 expression with TP53. And so that apparently there was a mutation in an upstream sequence about 26 or 30 million years ago, where a promoter region was very receptive to TP53 in elephants. And so TP53 strongly controls LIF6 expression, and that allowed for lower cancer incidence and therefore for elephants to sustainably, at a population level, generally adopt larger and larger body sizes. So very interesting how this is how elephants kind of surpass kind of the mutational burden that comes along with large body size by adopting greater and greater tumor suppressor symptoms, tumor suppressor functions with LIF6 and TP53. So I hope you enjoyed this video and learned something from it. And this is kind of a screenshot of the paper's title and recording. And so you can go back and read through and come to your own conclusions as well. So I hope you learned something. Thank you.